Next up, I would like to welcome Sabrina Taylor with the LSU AgCenter. She's going to be talking about everyone's favorite marsh bird, the seaside sparrow. Oh, and Sabrina is a keen gardener and is testing out a native species currently. Thank you. All right, thanks for coming, everybody. I'm going to talk today about, does the slide advancer work? No, use the... Talk about some work that I've done with my co-PI, Phil Stouffer, who is also at LSU with me, 3 postdoc fellows, a couple of PhD students, and multiple collaborators. So we're going to cover a lot of stuff, not necessarily in my area of expertise, because all these people branched out into lots of different areas of research. So we were funded by the Gulf of Mexico Research Institute. Phil and I are part of the Coastal Waters Consortium, which consists of 20 PIs plus or minus all over the country who are studying lots of different aspects of the marsh, from the sediment to the grass to the microbiome. And for Phil and I, the effects of the oil spill on seaside sparrows and marsh rice rats. So I'm going to cover the sparrows today since this is a talk on birds. Okay, so a little bit about seaside sparrows. They're dependent on the salt marsh. They're only found in the salt marsh. They are non-migratory, so they're there all year. So if there's an effect of oil, they're going to be exposed to it all year. They're also a top predator in the marsh, which is kind of interesting, even though they're quite a small bird. There's not a ton of species diversity in the marsh. What's there tends to be there in very high abundances, including this sparrow. Okay, another thing that happened while we were undertaking this study is that Hurricane Isaac came ashore in August 2012. So the track went right next to our study sites, which are in Barataria Bay here. We think that based on the storm surge hindcast, so this is showing that our study area was under something like 7 to 10 feet of water for several days. It displaced probably 500,000 to 1 million seaside sparrows during that time because all of the marsh vegetation was underwater. The birds had nowhere to perch, and so they went somewhere else. It probably also greatly affected their diet, right? Because the terrestrial insects probably got wiped out too. We have these birds banded, so we know that the next year many of them returned, sometimes to the exact spot where we banded them. But we just need to keep in mind during our analyses that Hurricane Isaac is probably playing a role here as well. Okay, so, right, hurricanes would be old news to seaside sparrows. These are the storm return rates along the Gulf Coast. And in Florida, every 3 years you can expect some type of a storm, a tropical storm up to a major hurricane. So the birds are probably adapted to moving around, but nevertheless it's going to probably affect our results on some level, the effect of this hurricane. Okay, so into the oil spill here, these are where our study sites were placed. So we had oiled sites in red, sites that were initially unoiled in blue, and then later on we'd added some reference sites on the other side of the Mississippi River where oil didn't make landfall. All right, so Deepwater Horizon oil spill that happened in 2010. U.S. Fish and Wildlife Service saw oiling on feathers in seaside sparrows, and then we started undertaking a series of studies. So we looked at oil in feathers and gut contents. We looked at CYP1A gene expression. We looked at microarray gene expression. One of our collaborators looked at diet using stable isotopes and fatty acids. I have a PhD student who's looking at diet with DNA metabarcoding. And finally, one of our collaborators and former postdocs is looking at how this all translates to reproductive success and abundance in the birds. So I'm going to go through these fairly quickly, and if you have any questions, you can get me during the panel. Okay, so carbon and oil. The way this works is that cosmic radiation interacts with nitrogen in the atmosphere, and that continually creates C14. And that type of carbon is absorbed by plants in carbon dioxide, and then those plants are eaten by other trophic levels on up in the food chain. They die. You can radiocarbon date, and this is how you tell how old a sample is, basically by looking at the ratio of C14 in the environment versus the amount of C14 in the bone or the organism or whatever it is you're looking at. C14 is a radioactive isotope, so it decays into C12 and C13. And so over time, you expect to see less C14 in things like, mummies and things like that. The way you can use it in oil spill research is that oil has been in the ground for so long that all of the C14 has decayed. There's none left. So if any of the organisms have incorporated that oil or that carbon from oil into their bodies, and I mean on a molecular level, then you'll see less C14 than you expect to see from a modern sample. Okay, like if you radiocarbon dated it, it would look like it was 500 years old or 1,000 years old or something like that. So we looked at, we looked at C14 in feathers in seaside sparrows, and you can see that we have less C14 in oiled bird or on oiled plots than on unoiled plots, which suggests that C14, so carbon from oil, was incorporated into the molecular structure of the feathers of the birds. We also looked at C14 in the gut contents of the birds, so the insect prey that they'd been eating, and there's also a smaller amount of C14 from birds on oiled sites versus unoiled sites. So probably the C14, the reduction of C14 you're seeing in the feathers was probably as a result of the ingested food that they took, which also had those lower levels of C14. Okay. All right, next study, CYP1A gene expression. So CYP1A is a gene that's involved in detoxifying polycyclic aromatic hydrocarbons, which are one of the major toxic components in oil. And what this gene is basically doing is producing a protein that helps make the pHs water soluble so that the animal can excrete that toxin. So if you have an oil spill and you have oil in the environment and it gets into a bird, you expect that gene to start expressing. So it's going to start making lots of copies and it's going to start producing those proteins. Okay, and the liver is really sort of your main detoxifying organ in the body. So it detoxifies alcohol, drugs. It also detoxifies pHs. So if you collect a liver sample from birds on oiled versus unoiled plots, you can look at the difference in gene expression of this gene to see whether or not they were exposed to PAHs. All right, so what are we seeing? So the expression of this gene is on the y-axis here, and the year is on the x. In 2011, there's a significant difference in expression between birds on oiled and unoiled plots. After 2011, there is no difference across the different plot types, oiled or unoiled. Hurricane Isaac hits in 2012, the following year when we collect our samples. So we collected our samples in June, and this hurricane hit in August. So it's really the 2013 samples where we expect to see a difference here. We see an increase in CYP1A gene expression on all sites, including the reference sites on the eastern side of the Mississippi River. So this is suggesting that the oil that was buried in the marsh got resuspended, moved around, and became something that birds were exposed to in a number of different areas. And there's other data from one of our collaborators who works on loons, who also saw an increase in PAHs in blood samples in the loons following the hurricane. So it looks like this hurricane stirred things up. Okay, so CYP1A higher in 2011 than on oiled versus unoiled. 2013, after the hurricane, was significantly different from other years, from 2012 and 2014. We also, quite a few people in our consortium were collecting sediment samples that were analyzed for aromatics to see what the concentration was. So the bars here are the concentration of aromatics. The dots is the CYP1A gene expression. 2011, there's also a big difference in the amount of aromatics we're seeing in sediment samples between oiled and unoiled sites. The other place we see a big difference is in samples that were collected in 2012 pre-Isaac as compared to samples collected in 2012 post-Isaac plus the 2013 samples. There's a big increase in oil in the sediments. So that matches well with our gene expression results, right? We're seeing more aromatics in the environment. We're seeing a higher level of gene expression in the birds. Okay, so that's just one gene. What about other genes? There are genetic techniques that you can undertake, like a microarray, which are going to look at something like 18,000 described genes in whatever tissue you're analyzing. So these are preliminary results. I'm just throwing in a couple of slides of preliminary results here. So in liver samples in the Seaside sparrows, these are 2011 samples, and we saw a big difference in CYP1A gene expression. We want to see what other genes are doing. There's around 270 other genes that are differentially expressed. And the way you can summarize these data, or one way to do that, is to look at pathway analysis. So genes aren't usually working independently on their own. One gene makes a product that, that sets off another gene, and you have this whole pathway. So you can lump genes together that are part of the same pathway to see which major pathways are affected when birds are exposed to oil. So in the case of the Seaside sparrows, we're seeing pathways associated with liver damage, liver proliferation, and cell death being expressed, and also cardiac enlargement, which goes well with the fish data, where fish have been experimentally exposed to oil in tanks. You see issues with cardiac tissue, and also, of course, the presenter just before me discussed this in birds as well. So even though this is liver tissue, we are seeing differential expression of cardiac genes happening. Okay, so moving on to a different angle here. Now we're looking more at the food web. So this is data that Jill Olin and Mike Polito analyzed. They're looking at stable isotopes and fatty acids. This lets us look at how the food web may have changed. So we know that the invertebrate populations responded to oil. So the terrestrial food web may also have been simplified in oiled marshes relative to unoiled marshes. And the Seaside sparrows that are using oiled marshes may consume different prey compared to the unoiled marshes, depending on which prey survived the oil and which prey didn't. We expect both of these differences, if they exist, to weaken over time. Okay, so just a little bit of background about stable isotopes and fatty acids. Here, basically the idea is you are what you eat, so the kind of food you're eating is going to be reflected in your tissue, right? So bulk stable isotopes, carbon is looking at production resource. So this is looking at the type of carbon that exists low down on the food web. So whether it's an aquatic source or whether it's a terrestrial source like Spartina. So you can differentiate between those two types of carbon. The other isotope you can use is nitrogen, and this lets you look at the trophic level. Okay, so higher levels of the trophic food web are going to tend to have higher amounts of this isotope of nitrogen. The third type of tracer you can look at are essential fatty acids. These are acquired only through diet. They indicate food quality, and they also will help you distinguish between whether the trophic web is using an aquatic versus a terrestrial source. Okay. So as we go up, oops, no, wrong way. As we go up the food chain here, primary consumers, secondary consumers, tertiary consumers are all going to have different amounts of nitrogen. So we know based on the amount of nitrogen what level of the trophic web they're occupying. Okay, so what are the results here? So that top graph is looking at the mean carbon stable isotopes. The graph on the bottom is looking at the mean nitrogen stable isotopes. There's no differences between oiled and unoiled sites for either of these elements. There is a significant temporal difference for nitrogen in 2013. So this is also following Hurricane Isaac. And what this is telling us is that the feeding level is at a lower trophic level in this year. And this is potentially because those marshes were completely inundated with water, which possibly wiped out terrestrial insects like grasshoppers, say. And so that type of prey needed some time to recolonize the marsh before they were present again and before they could become prey to species like the seaside sparrows. The fatty acid profiles, so this is a PCA that's showing the different years here. 2013 is clustering things separately from the other years. So there's this significant temporal difference in 2013 here as well, like the nitrogen. And if you look at the fatty acid sources, so the bottom in orange, that would be a terrestrial resource that's based on Spartina. Farther up in blue, those are fatty acids that are associated with a benthic source, so benthic diatoms and bacteria, for instance. So 2013 was characterized by a high proportion of benthic aquatic-derived resources relative to other years, which also suggests that the hurricane had a big effect on which resources were present in the marsh for sparrows. No significant differences between oiled and unoiled plots for the fatty acids either. Okay, so it doesn't look like the oil spill is really affecting the trophic food web here. Another approach we took was to look at seaside sparrow diet. So what are the sparrows eating specifically in terms of species across these years in response to potentially altered prey communities? So what we're doing here is we're taking fecal and gizzard samples, we're extracting the DNA, we're aligning those sequences to a reference genome to find out what insects are in their diet. So the preliminary findings here, their main diet taxa seem to be crabs. So this could be crab eggs because the DNA is not going to distinguish between juvenile forms and adult forms. Interestingly, we've also caught squareback crabs eating seaside sparrow eggs, so they got their own back here. Spiders, especially wolf spiders, moths and butterflies, we're still identifying these ones. Unidentified arthropods. Interestingly, with the hemipterans, so these are the true bugs, the insects that are sucking plant juice, basically. There were very low abundance from 2011 to 2014. They really started increasing from 2015 onwards. So lots of them around after that. Alpha diversity, this is telling us how many species are there. It doesn't really distinguish between which ones. So 5 spiders versus 5 crabs, you'd still basically get the same metric with alpha diversity. There's no difference among oiled and unoiled sites. Years are not significantly different, but 2013 looks different. We need to finish the analyses here. 50% of these reads are unassigned, and it's just because we need to add more sequences into our reference genome to sort this out. Beta diversity is considering which species as well as how many, so you can look at differences in shifts in community with this. 2011 to 13 were significantly different from each other in all other years. In the years afterwards, that stabilized. Okay, so how does all of this translate up to population level stuff like reproductive success and survivorship of the sparrows? So just because they're expressing CYP1A, for instance, does that mean we're seeing big difference in abundance and reproductive success? Abundance, you might think that there was a recovery from 2012 onwards until you collected data in 2017 and saw those abundances going back down again. So here, really, we have a lack of baseline data, and whether it's oiled or unoiled changes depending on the year. What about reproductive success? So we're following nests, hopefully from initiation through the egg stage, through the nestling stage. Here, what we're finding is most nests tend to fail. They're depredated from mink, marsh rice rats, raccoons, very high nest failure rates generally. Always higher on oil plots, but there are other confounding variables here, right? So these plots tend to be a bit drier, the oiled ones do. There may be more predators present. There's no difference in marsh rice rat abundance. We have those data, but we don't know about other species like mink and raccoons. Okay, so to sum up here, sparrows post-oil spill, they incorporated oil from carbon into their tissues on oiled sites. We saw an upregulation of CYP1A, which detoxifies PAHs, as well as lots of other genes on oiled sites. Stable isotope and fatty acid profiles unaffected by the presence of oil. Diet with DNA analyses unaffected by oil. Do these effects translate into population level effects? There's this trend of higher nest failure on oiled sites, but we also have a lot of confounding variables there, so we can't really nail that down. Post-Isaac, we also saw upregulation of CYP1A on all sites, whether they were initially oiled or not. We saw a lower trophic feeding level and more benthic aquatic-based prey in 2013 following the hurricane. And the diet betta diversity from 2011 to 2013 was significantly different among years and stabilized after 2014. So I just want to say thank you to all the people who helped with this, especially our 25 plus field assistants who were pretty stoic working in these marsh conditions. Thank you.